Good evening, class. Good evening. Just join in. I have some classes. I just finished at 7:30, so I just need to get freshen up. That's why I got late. Sorry for that. Mm. So today we are dealing with the two topics. That is protoplast isolation and cybridisation. So that will be the main two topics we will be concerning with today. Just join in, everyone, join in. More students to join, yes? Or we have that much only today? So I'm receiving your assignments today. That's good. That's very good that you're working for it. So I should start now, I guess. Let's start with the protoplast isolation. And regeneration. So mainly in the protoplast isolation and regeneration is the happening to the cells which doesn't have cell wall, and inside that whatever is left with the mesophyll cells, that is the mesophile cells rest, and then we use them to to mix them to different species, intergenic species, that is known as your yeah, somatic variation or somatic hybridization. So that's why that's the main difference. So in this topic, we will understand how you can isolate your protoplast with different techniques via mechanical and enzymatic method, and further how we could use it to learn for our for the purpose of plant biotechnology, how we could serve the purpose of plant biotechnology. That will be dealing in this topic for today. So this is protoplast isolation, culture, manipulation, and fusion. All these topics will be covered today in this topic. So this is the appendix of it. So mainly we deal with the introduction, some isolation, its culture, its development, and regeneration. with the protoplast. So the term protoplast was first given by Einstein in 1880. It refers to the cellular containing excluding cell walls, or can be called as naked explant cell wall. So it was described with the help of scientist Kleckers. First time he carried out in 1892 using a mechanical method on the plasmolyzed cells. So the application was were quite famous at that time, undergo fusion under the help of genetic engineering and crop improvement. The experiments mainly involved three stages, that is protoplast isolation, protoplast fusion, leading to gene uptake of the two different plants, and then development of the regenerated fertile plants from the fusion products. So whatever you are working with, so it helps to develop into regenerated fertile plants from the fusion products. That is your hybrid product is produced by this case. So let's go next. Um, Further, in the isolation process, as explained, it could be of two type, that is mechanical and enzymatical. Mechanical is somehow you lose your most of your plants, your most of the cells will be lost, yield will be not so good. But enzymatical, on the other case, is quite high advantages. It is always preferred because of proper yield and lower tissue damage, while mechanical method causes maximum tissue chopping with lower protoplast yield. So both of these methods we will discuss now. First is the mechanical method. It is uh, given by the Clerkel in 1892, pioneered with the protoplast by mechanical method. So in this method, what he did, he kept his suitable plasmolyticum, for example, with the CPW containing 13% weight by volume mannitol. Mannitol was used as a washing media. Uh, to remove the undesirable things. So once the plasmolysis is completed, while remaining in the osmoticum, the leaf lamina would cut with a sharp edge knife. So while remaining in the osmoticum, that is your mannitol, plasmolysis is completed. So from the leaf lamina, you cut it with a sharp edge knife. Then after cutting with it, in this process, some of the plasmolyzed cells were also cut through this cell wall. So releasing the intact protoplast, while some of the protoplast may damage inside many cells while cutting with the own hands. So this could be sometimes damaging to our plant cells, so which is not a good way to go for. So that's why protoplasts were damaged in this cell, and the released protoplasts then have to be separated from the damaged ones and cell debris. So you need to have some separation thing at the end of it, uh, of your experiments. So, what are the disadvantages of using mechanical methods? That is lower protoplast yield, labor intensive method. Protoplast obtained has low viability. Method is applicable only to the vacuolated cells. Further, then comes the enzymatic method in the protoplast isolation by E.C. Cocking in 1960. He demonstrated for the first time that enzymatic isolation of a large number of protoplasts from cells of higher plants. So this method involves leaf sterilization followed by peeling of the lower epidermis to release cells which are plasmolyzed and added to enzyme mixture followed by the harvest of the protoplast. So this has been shown in this figure 12.1. Here we can see that first is your explant, from that we took the leaf, and this is the explant B, from there we took also the leaf. And then from these parts we remove the cell wall by using enzyme cellulase enzyme. By using the cellulase enzyme, we remove the cell wall. Then from both of them, we isolate the protoplast. And then protoplast is fusion as per this method with the 1, 2, 3, 4, 5 stages, which leads to the microvilli. Microvilli will produce callus, then regenerated callus, and then plantlet. So these are the steps involved in the protoplast isolation, fusion, and regeneration. So this is the main step that has been done for the protoplast isolation. So as we can see that this enzymatic mixture could be of can be used sequential enzymatic hydrolysis and mixed enzymatic hydrolysis. In sequential isolation, firstly the cells are separated by the use of maceration enzyme. That is your pectin hydrolyzing enzyme such as macrozyme or the pectolase. So once the cells are separated, they wash in the CPW solutions free of enzymes but containing plasmaticum, and also by gentle centrifugation at 100 G. The palate is retained and is suspended in the second enzyme like cellulase and hemicellulase. So the palate is then put up in the second enzyme called as cellulases and hemicellulases, used to hydrolyze the remaining cells that has that component. So try to remember these enzymes, what are their functions are. That is the main thing that you have to remember in the protoplast isolation. So once the protoplasts are released, they are washed with CPW to remove the debris. So all the debris are removed in the sequencing isolation. In the mixed enzymatic approach, what you do is plant tissue cells are plasmolysed in the presence of mixture of enzymes, that is pectinases, cellulases, thus inducing simultaneously separation of cells and degradation of their walls to release the protoplast directly in the cell single step. So steps involved in the protoplast isolations includes fusion and regeneration. So this is I have already explained to you isolation, fusion, and regeneration, how it is all done in a very nice and simple manner. So what are the conditions that are required for the enzyme activity? So basically, you need enzyme have to be taken care by checking up its pH and temperature, because enzyme are highly dependent on pH and temperature. Thus, for enzymatic release of protoplast, enzyme showing activity of pH range 4.7 to 6 and temperature range from 25 to 30 degrees Celsius is used. So duration of enzyme pre-treatment and condition of light presence required for incubation may be also determined. So duration of enzyme pre-treatment and condition of light presence, how much do you require, it could be also determined in this method. So enzyme mixture used should essentially consist of cellulose, hemicellulose, and pectinase. These three main enzymes that we used for the protoplast isolation, and which facilitates degradation of cellulose, hemicellulose, and pectin respectively. So cellulose for the cellulose, hemicellulose for the cellulase for the hemicellulose, and pectinase for the pectin to degrade these enzymes in the protoplast. So hence the concentration of sugar alcohols used as osmoticum must be empirically defined. So you have to check how things are working out, how things are done. You have to identify them as it is. So factors affecting yield and viability of protoplast. So factors which are affecting yield and viability of protoplast are as follows. So source of material, that is uh, leaves were the most convenient source of plant protoplast because it's allow the isolation of large number of relatively uniform cells without killing the plant. So without killing the plant, from the leaf we can get large number of protoplast in a very quite easy manner. Very convenient source of plant is. Moreover, the mesophile cells are loosely arranged. Enzymes have an easy access to cell wall. Mesophile cells are the best one to go for these experiments, and the parent plant age and the condition in which it is growing have profound effect on the yield of protoplast. So due to the difficulty in isolating culturable protoplast from leaf cells of cereals and some other species, cultures can be used as a source of material. And the yield of protoplast depends upon the growth rate, growth phase of the cells. So yield that is being produced, it's mainly depending upon the how much is the growth rate and growth phase of the cells. Generally, embryonic suspension culture are used to obtain a totopotent protoplast. So we use ESC culture, embryogenic suspension culture, to obtain a totopotent protoplast to find the answers for that. Then that was something about we have done the factors that are affecting the viability of protoplast. Before that, we dealt with the conditions that are required for the enzyme activity. So first we dealt with the source of material which could affect the protoplast isolation. Second is the pre-enzyme treatments, that which kind of enzymes we are treating it. So to facilitate the penetration of enzyme solution intracellular spaces of the leaf, which is a very effective digestion, various methods are followed. The most common is to peel the lower epidermis. You remove the lower epidermis and float the striped pieces of leaf on enzyme solution. Let it float on the enzyme solution flow. So in manner that peel surface is in contact with the solution, when combined with the vacuum infiltration isolation, the later approach has proved very effective. And then you make a vacuum infiltration to let it be consumed by the whole enzyme of your leaf, and hence you can have your pre-enzyme treatment could be good. Then afterwards, enzyme treatment is quite important, which factors make your protoplast isolation to be very rigorous. That is uh, the release of protoplast is very much dependent on the mature and concentration of enzyme used. The two major enzymes required for the isolation of protoplast are cellulase and pectinase. So what are the two protoplasts that they are using? They are using cellulase and pectinase. The cellulase is required to digest the cellulose cell walls, and pectinase mainly degrade the middle lamella. So some of the tissues may require other enzymes like hemocellulase, diacylase, macrozyme, and pectolase also. The enzyme activity is pH dependent, and most of them are adjusted somewhere between 4.7 to 6 pH. 4.7 to 6 pH, they are being arranged in that order. So choice of plasmolyticum is the two most common used compounds are sugar alcohols, mannitol, and sorbitol. So these are the mainly the sugar alcohols, the plasmolyticum that we used in our case. Of these, mannitol is the most preferred since it is not metabolized by the plant cells. So once the protoplast device regenerates the cell wall, no more osmoticum is required. So it is therefore should be removed gradually from the medium, otherwise cell division stops. To slowly remove the osmoticum from the medium, the protoplast can be isolated in high osmoticum mixture consisting of both mannitol and sucrose. The sucrose will be metabolized by dividing the protoplast and thus will reduce the osmolarity of the medium. So normally mannitol is used at higher concentration of range of 1 to 11 to 13 percentage, and solution into which osmoticum is often but not always added is salt mix called as CPW salt mix, CPW for short. So this has been observed beneficial rather than using the distilled water instead of using the high yield of viable protoplast. So just remember basic parts that why which are the highest that most of the things that we used in our plasmaticums, that is the sugar alcohols, mannitol and sorbitol, and the concentration ranges from 11 to 13%, and we have to maintain a quite high level of it. And basically the best mixture is containing of both mannitol and sucrose to remove the osmoticum from the medium. So this is the salt mix of protoplast washing media. So this is was done by Cocking, Pavardy and White, the three scientists. That's why CPW media. So this is a washing media which has KH2PO4, KNO3, CaCl2, MgS4, potassium iodide, and copper sulfate, CSO4. So they are made in 1 liter. Like this much, if you add, then your CPW is ready to use, and you can use it for your further experiments. So once you did the isolation, now it comes to the purification. So enzyme treated results in suspension of protoplast. Undigested tissues are like cell debris and cellular debris, undigested tissues. The suspension is passed through the metal sieve or a nylon mesh in order to remove the undigested cellular clumps. The filtered protoplast enzyme solution is mixed with a suitable volume of osmoticum. Solution is centrifuged to the pallet the protoplast, and pallet of protoplast is resuspended in the osmoticum of the similar concentration as used in the enzyme mixture. So this diagram is showing us in figure 12.2 that how the protoplast purification is done. So here we can see that first you take a leaf or friable callus for protoplast isolation. Either they take the callus or the leaf, and then leaf or the hard callus are chopped into small parts and mixed with the enzyme solution and the osmoticum. So we add the osmoticum that we have discussed like mannitol, sorbitol, or and also the enzyme solutions like cellulase, hemicellulase, pectinase, and then we incubate for the 16 to 18 hours. For 16 to 18 hours we incubated that. Protoplast settle at the bottom of the dish, and enzyme solution is removed. As it is settled at the bottom of your dish, so enzyme solution is removed. Then protoplast transferred in the washing medium. Then they are transferred into the washing medium like this. uh, then they are centrifuged to at the bottom of it. Then they're resuspended in the sucrose washing media. Then you have debris at the bottom and protoplast obtained after centrifugation at the top. and then you resuspend into the culture media, and then you remove small sample into a hematocytometer. So there are four chambers like here. So what you do is you calculate in the one chamber how many cells are, then you multiply by the four chambers, then you have an average of it. Then you calculate the average, like how many you calculate in all those chambers, one, two, three, four, and then at the end you calculate an average, like how many cells you have. Then you can calculate in per ml of your solution of your experiments. So you remove small sample and place it in hematocytometer for protoplast count, then resuspend the culture in the medium to give pre-plating density. Then you add into the plating density the how much cells that you require to grow. Then you can go for a further experiment later on. So that's how you do your protoplast isolation. No, sorry, purification. This was for the purification. Isolation we did in previous diagram. This is for the further purification of a sample. So how to, and also your protoplast viability, how could this be factored depending upon your protoplast isolation. So the isolated protoplast must have spherical shape, so we have to check whether we having spherical shapes or not when observed under the light microscope. This we could check with the various staining methods, that is fluorescent diester staining method, also chlorophlore white staining method, protoplast viability, variation of protoplast size. So these are the four different methods you can check of a protoplast isolation. So FDA accumulates inside the plasma lemma of viable protoplast, and live protoplast contain esterases which leaves FDA to release fluorescein, which fluoresces yellowish green using fluorescein microscopy within the 5 minutes. So FDA dissociates from the membrane after about 15 minutes, and it is used as a concentration of 0.01 dissolved in acetone. Whereas the calcifor white staining, this staining method assures protoplast viability by detecting onset of cell wall formation. So calcifluor binds to a beta-linked glucosides in newly synthesized cell wall, which can be observed as a fluorescent ring around the membrane. So optimum staining which is required is 0.1 ml of protoplast mixed with a 5 microliter of solution of a CFW. Further, protoplast viability could be detected using oxygen uptake by having oxygen electrode, which could tell us how much respiration is happening in our system. And also variation of protoplast size with changing osmotic concentration also enables to viability of the protoplast. So by all these four methods, you can check the protoplast viability, how much it is going on. So there are, now we're gonna discuss further. So students, you will never find anywhere, it's being, these lectures are being arranged with the help of HS Chawla also, and also from the notes from AIR. So these notes you will never find anywhere, so they are quite inclusive and intensive notes for you. So try to go through them once, and they are the best of the best notes you can ever see. Go through all any other institute or any YouTube videos, you will never found these intensive notes actually. So I did thorough research before going through this, because I thought there are a lot of literature is going with you. It might be difficult, but I found this to be really on the other side positive actually, because this much of intense knowledge was not present in such a nice manner, in a nutshell, of all about the protoplast culture, or somoclonal variation, or somatic embryogenesis, or synthetic seeds, or hybrids. Everything that we studied so far is very precise and good to go. So what you're getting is getting the best so far from the plant biotechnology perspective. So let's continue with that. The protoplast culture techniques, their culture requirements and the culture method are same for both protoplast and single cells. Main difference is the suitable osmoticum. So what kind of suitable osmoticum we required to regenerate a strong wall. And to do so, sometimes protoplast is first grown in the liquid media and then transferred onto the agar media plates. The following techniques have been adopted in order to maintain number of protoplast population between minimum and maximum effective densities after plating up. So the first is your liquid method. This method is preferred in earlier stages of culture as it provides uh, easier dilution and transfer. The osmotic pressure of liquid media can be effectively produced after a few days of culture. the cell density can be reduced, or cell of special interest can be isolated easily. So all these things could be done with the help of liquid method. And liquid method also, the protoplast suspension is plated as thin layer in the petri plants. So we use also stored in the humidifier chamber at the end of it. Or you can, instead of liquid media, you can also use agar or agarose media. So agarose is preferred choice in place of agar, as this has improved culture response. And also agar culture keeps protoplast in fixed positions, thus prevents it from the forming clumps. Further, immobilized protoplasts give rise to clones, and which can then be transferred to the other media. So, in practice, the protoplasts suspended in the molten agarose medium are dispensed into small plates and allowed to solidify. An agarose layer is then cut into four equal blocks and transferred to the larger dishes containing the liquid media. So third point is the feeder layer. So in the feeder layer, we have a low density of your culture protoplast, and feeder layer technique is adopted when you have low density of a protoplast. So feeder layer of X-ray irradiated non-dividing but metabolically actively protoplast after washing are plated in a soft agar medium. And then non-irradiated protoplast of low density are plated over this feeder layer. Protoplast same species difference can be used as a feeder layer then. Then in co-culturing, this method is done to do the co-culture of protoplast, two cultures of different species to promote the growth of the hybrid cells so that the growth could be done faster. So metabolically active dividing protoplast are of two types, slow and fast growing culture. The fast growing provides the other species with the diffusible chemicals, growth factors, which help in the cell wall formation and cell division. So that's why the fast growing is helping the slow growing by providing them the cell wall formation and cell division. Whereas a co-culture method is generally used in the calli, in the callus culture, arising of the two types of protoplast, morphologically different. For example, we use this method for albino plant and green plant. For the albino plant, we use it, and in the green plant are easily distinguishable based on color, where albino protoplast will develop non-green colonies. So this is showing you that those four methods, four different media that you're using to grow your protoplast. First one is then the liquid medium that we discussed recently. Protoplast is, you can see, growing on the top, and the bottom is the liquid media. Then in the solid media, that is your agarose media they prefer. You have a protoplast and semi-solid media at the bottom. Then with the feeder layer, that there is a feeder layer in the top, and the bottom is the media. Then on top is the protoplast. Then there is a slow growing protoplast and the fast growing protoplast in the co-culturing of slow and grow protoplast technique. So the fast one, they are providing the cell wall and regeneration to your slow one also. So bottom is the medium. So these are the four different types of medias that you can use to grow your protoplast also. And what about the culture medium? What about the culture medium? How it should be? It should be nutritional, first of all, yes, similar to the culture plants that we are doing, mostly salts of Marishi and Skoog media that we did in the practical part, right? And also B5 media, and their modifications have been used also. So in this media, we need ammonium salts are quite detrimental for the protoplast survival. Concentration of zinc is reduced while concentration of calcium is increased. It enhances the membrane stability. Osmolarity is maintained by addition of sorbitol. Osmolarity is increased by increasing the addition of sorbitol, mannitol, glucose, or sucrose, and mannitol being widely used in the osmoticum and is used by the dividing cells. Glucose is preferred in this case, carbon source as a source. Sucrose do not satisfy protoplast culture. One or two amino acids are added at low concentration. Growth regulators such as high concentration of auxins NAA240 along with lower concentration of cytokinins is used, ABAP and zetin. Environmental conditions are quite important while growing it. Is high light intensity inhibits the growth of protoplast. So initially we have a dim light for a few days, then transfer to the 2000 to 5000 lux. However, it's better to grow them in the darkness actually. So light is there, hormones are there, glucose is there, osmolarity is there, concentration of zinc is matter, ammonium salts are matter, your medium is matter. So all these matters while you are culturing your protoplast culture. Then comes the plating densities. Then you just plate uh, your protoplast culture. So like cell culture, the initial plating density of protoplast has profound effect on the plating efficiency. So protoplasts are cultured at the density of 1 is to 10 is to power 4 to 1 is to 10 is to power 5 protoplast milliliter of the medium per milliliter of the medium. So this much is more than enough. At high density of the cell colonies arising from the individual protoplasts tend to grow into each other, resulting in the chimera tissue if the protoplast population is genetically heterogeneous. So cloning of the individual cells, which is desirable in the somatic hybridization and mutagenic studies, can be achieved if the protoplast or cells derived from them and can be cultured at the low density. So protoplast development and regeneration. So we did isolate it, purified it. We take care of the culture media. We take care of its various layers. How we have to do it. We have checked by the various factors it has been affected. This protoplast isolation now comes to check its development and regenerated to the whole plant. It's comes the last stage of it. So in this case, protoplast starts to regenerate a cell wall within few days of culture, and during this process protoplasts lose their characteristic spherical shape, which has been taken as the indication of new wall regeneration. Cell wall regeneration can be confirmed by the calcoflow white staining method, and there is a direct relationship between wall formation and cell division. And moreover, protoplasts which are not regenerated a proper wall fail to undergo normal mitosis. Further, protoplasts poorly developed wall often show budding and may enlarge several times than the original volume. They may go multinucleate because of karyokinesis, which is not accompanied by the cytokinesis. So karyokinesis is going on, but cytokinesis is not going on. So there could be multinucleation is possible. Among other reasons, inadequate washing of the protoplast prior to culture could lead to abnormalities. And furthermore, a complete process suitable conditions such as light, pH, temperature for the newly synthesized protoplast are quite essential and can be visualized by staining. So when the cell wall formation is completed, cells undergo division, results in increased size of cells. After an interval of 3 weeks, small cells clonies appears. These clonies are transferred to osmotic-free callus induction medium, and this is followed by introduction into organic or uh, embryonic medium. Yeah. So these are your protoplast isolated cell wall regeneration. You can see on the left side an isolated showing spherical structure. So they are in the spherical structure your protoplast, and on the right side is the wall is being generated, and also they started to grow with a cell division like this. So that's it from the protoplast part. Thank you very much. Uh, that's enough for this from this point of view. Any questions, students? Any questions? If not, let's take a 10 minutes break now. Then we start the next part, and I hope we will finish it early today. It will not take much time. So I will take now your next part of cybrids, and so this topic, somatic hybrids and cybrids. This is also very interesting topic. Uh, we will deal with this part also today. So let's meet in 10 minutes. It's in my watch is 40. So let's meet at 10:50 then, around 10:50. And we start with this topic. Products of somatic hybrids and hybrids. So students, welcome back. Come back now also. We start with another topic of products of somatic hybrids uh, and what are the little bit of the applications in crop improvement, how we could use them to improve crops actually. So we discuss with this part now in the later part of our section of this. Um, So products of somatic hybrids and cybrids. So this topic, basically somatic hybrids are the one in which there's one mitochondria is used, in other case cytoplasm is used. Then you fuse them together of two different species like tomato and potato, and then you create a new species out of it. And that is your somatic hybrids. And cybrids are the one which has um, this thing called on your seats, they are more into other side of it, so we will understand more about them in upcoming lectures. So appendix, so first we study about the chromosome status of somatic hybrids, then practical applications of somatic hybridization and cybridization, then comes the somatic hybrids for the cytoplasmic male sterility, then comes the generation of the cytoplasmic male sterility by fusion, then cytoplasmic transfer. So what happened at the chromosome level in the somatic hybrids? They are mainly cytologically, they become diploids, that is amphiboloids we call them, amphiboloids. That is, there's number of chromosomes within the nucleus, they become double. So chromosome number of somatic hybrids are successfully obtained through the protoplast fusion. So these are the stages I'm telling you after the protoplast fusion is done, isolation is done, purification is done. So and what is made is afterwards is your somatic hybrid, and it's now telling us about the latest story about it. So it has exact number expected, and it's called as amphiboloids, that is a diploid number of chromosomes, and hence selection will also need to be applied at a cytological level. If true, amphiboloids need to be obtained the variability in the chromosome number in hybrids due to the one of the following reasons, that is multiple fusions give higher chromosome number, and in PEG induced electron induced fusions between more than two protoplasts is possible. So one of the reason for the variability in the chromosome number in hybrids is also asymmetric hybrids, that is fusion from the protoplast isolated from the actively dividing tissues of one parent and quiescent tissues of the other parent. So unequal rates of DNA replication in two fusion partners may also give rise to asymmetric hybrids. Whereas somoclonal variations in culture cells used for the protoplast isolation may be also lead to the variation in the chromosome number. So further, a practical application of somatic hybridization and cybridization. Uh, so first is mean of genetic recombination in asexual or sterile plants. Uh, so you can have a very genetically recombinant and also sterile plants because of this at the end of your stage. So somatic cell fusion appears to be the only one approach which have approached through which two different parental genomes can be recombined among the plants that cannot be reproduced sexually. So two species which are not possible in nature, so we can grow them here with the help of somatic hybridization. Further, protoplasts of sexually sterile, that is your haploid, triploid, and aneuploids plants can be fused to produce plant fertile diploids and polyploids. So there are several reports describing the amphidiploids and hexadiploids plants produced from the fusion of haploid and protoplast of tobacco. So protoplasts isolated from the dihaploid potato clones have been fused with isolated protoplasts of the Solanium brevidens to produce hybrids of the practical breeding value. Further, haploid protoplasts from the anther-derived callus of rice cultivars upon fusion also produce fertile diploids and triploids hybrids. What are the overcoming barriers of sexual incompatibility? In plant breeding, sexual crossing at interspecific or intergenic level often fails to produce hybrids due to the incompatibility. So, in the sexual hybridization may therefore overcome by Somatic cell fusion. In some cases, somatic hybrids between two incompatible plants have also found application in industry or agriculture. So Scheider in 1978, he obtained ampho-diploid from Datura inoxia, discolor, and Dinoxia by fusion of Datura inoxia and Datura discolor, and also Datura inoxia with Datura stramonium. By fusing their diploid mesophytes protoplast. These hybrids do not exist in nature as conventional breeding procedures proved unsuccessful. Somatically produced amphidiploids of these combinations of Datura species are propagated for industrial use as they demonstrate heterosis and higher scomomelane content than in the parental forms. And further, Nicotiana repanda, Nicotiana nasophila, Nicotiana stockoni are resistant to a number of diseases but are not sexually crossable with tobacco. However, fertile hybrids have been reported in combination with the um, and Nicotana tabacum and Nicotana nisophila and Nicotana tabacum and Nisophila stockini by protoplast fusion. So using this approach of tomato or hybridized has resulted in synthesis of hybrids which are fertile somatically with a number of wild species used in breeding programs. And also interspecific hybridization involving the species that are sexually incompatible with the eggplant has also been resulted in the production of amphidiploids with a trait resistance to the verticillium wilt. So we have done so many things in the field of science, in the field of agriculture, produce so many species with the help of somatic hybridization. Also, rape seeds, also one of the example, napas, Brassica nigra, they are all Brassica junicea, they are all hybrids examples. So they had quite improvement and increased rootstock of the potential. So somatic hybrids for cytoplasmic male sterility methods were developed to substitute the nucleus of one species into the cytoplasm of another species whose mitochondria were inactivated. So in this case, so what they do in the method of producing a cytoplasmic male sterility in your somatic hybrids, so what they do is they substitute the nucleus of one species onto the cytoplasm of the another species whose mitochondria were inactivated, and this type of substitution led to generation of the cytoplasmic male sterility. So you have a male sterility in the cytoplasm generations that you produce, and for this purpose two types of protoplast are used, that is mesophyll protoplast of tomato and mesophyll protoplast of solanum aquilae, which are treated with idostamate to inactivate the mitochondria, and in second case they are irradiated with the gamma or x-rays to inactivate the nuclei. So in both cases we inactivate the nuclei with the help of mitochondria and gamma or x-rays respectively. So then protoplasts are mixed 1:1 ratio of your tomato and solanum aquilae. And then they are mixed in 1:1 ratio, induced to fuse calcium and PEG are added into that, leading to the production of heterologous or alloplasmic hybrids. Among the fusion products that are produced here, some hybrid tomato plants were indistinguishable from the original cultivars with respect to morphology, physiology, and chromosome number, but exhibited various degrees of male sterility. So we are successful in producing the male sterility into them. So in 5 tomato cultivars, male sterile induced into this manner was inherited maternally over the several generations. So for several generations, these cultivars, for 5 out of them, were cultivated like that. Therefore, it was obviously to see the cytoplasmic male sterility. So the mitochondrial DNA of these CMS hybrids did not resemble the mitochondrial DNA of their either plants and was instead recombinant type. So we produce a recombinant type in this case, presenting a hybrid mitochondrial genome. Therefore, protoplast fusion can effectively used for prediction of the CMS lines and has the following advantage. Only one step is required in this case. Nuclear genotype of the cultivar remains unaffected. There are prospects that 100% of progenies of somatic hybrids will be CMS, and the restorer of the CMS line will also shown to be available in tomato. So these are the various advantages of producing cytoplasmic male sterility, and this was a production method to do so. So this is how it is shown to you now. So you have your one plant here, tomato, another one the solanum. So in the first case of you damage the mitochondria in this case. So with the idone estamide. In the second case, you inactivate the nuclei by gamma or X-rays. In the second case, you inactivate the gamma with the gamma or X-rays. Then mitochondria, then you fuse them together with the mitochondria are fused of both of each of them and chloroplast. Then in the fifth step, you add calcium plus PEG, and there is a protoplast fusion of both happening now. Then in the seventh step, what is happening is chloroplast from the potato or as a protoplast being added to it, and also the nucleus from that part is being removed from that. So then at the ninth, you have nucleus, tomato nucleus in your new species, recombinant mitochondria, chloroplast mixture of both, fused protoplast, and somatic hybrid plant CMS resemble tomato. So 13 at the end, you have a full somatic hybrid plant resembling the tomato at the end. So that is how it's a bit complicated, but this is how it is done by removing the damaging the mitochondria first and removing the nuclei of the second, and then you bringing them together and joint them so that you have a recombinant mitochondria and also the cytoplasm of the both species, chloroplast, sorry, of the both species. So, Power et al. First, he found out that mesophyll protoplast of Petunia was cultured with the cell protoplast with the crown gall of Parthenicus and selected line of which were transformed to the Parthenicus but exhibited some cytoplasmic Petunia for some time. So this was followed by direct application of cybridisation in agriculture biotechnology by transfer of the cytoplasmic male sterility from these species to the P. axillaris. Besides that, cytoplasmic sterility, male sterility is a code that is used to check the rate of photosynthesis, lower high rate of temperature tolerance, and also resistance to disease and herbicides. So recent experiments on hybridization have been resulted in plants with reconstructed cytoplasm, combining mitochondrial DNA and cytoplasmic DNA and coded traits from the both parents. So the best example to show the potential for protoplast fusion in the reconstructed cytoplasm for practical purpose is the genus Brassica. So two desirable coded for cytoplasmic genus have been genetically manipulated through the interspecific hybridization between different species of Brassica. These traits include cytoplasmic male sterility and resistance to the artesa and herbicides. Further, CMS gene in Brassica plants, Diplotaxis muralis, Raphanus stavis is of the alloplasmic origin. And Raphanus stavis is of interest because it leads to the complete male sterility, and CMS restorer genes have been introduced into the rhapsid from these plants. Further, researchers in the past decades have shown plants can be raised in vitro and isolated plants, ranging to the angiosperms of species. So these are the various examples they are telling us how various species are done. I will cross them over. So this is interspecific hybrids produced through the protoplast fusion. So Brassica oleracea, Brassica campestaricis. So chromosome number of hybrids, wide variation we saw in them. So bringing like two different species, Brassica napus and Brassica oleracea. So it one has 18, one is 38, and one has 2n is 18. But still we are crossing them and we see wide variation of it. So like this way, for tobacco, for mesophile, for tobacco, for these are for all tobacco crossed with a different species of tobacco and having a different chromosome number, and you have 72, 96, so big number of chromosomes in your species. So you can see that how this hybridization is working out and how good it is. So the species which were not able to cross with each other, we are able to cross them, and easily we could grow with it. Things could be grow with them. So these are the various examples, various applications of hybridization in the field of plant biotechnology. And this earlier was interspecific, like interspecies, and this is intergeneric. Even between different genus, you can do the crossover, like Raphanus satavis between B. oberacea, oleracea, napus, and Aratica stavia. So you're producing new genus at the end. New genus is being produced, a new, totally new genus after crossing between two different species. So these are the various examples of them. And also intertribal between different tribes of somatic hybrids produce familia brassica. So this is totally new family has been produced with Arabidopsis thaliana cross with the Campesttaris to produce Arabidopsis arabidobrassica, also Thalpsobrassica and Barbidobrassica. With these different with the Brassica napus, you cross them with the Thalspi perfoliatum, Barberia vulgaris. So you cross them and you get your species as you look for. So indeed, these genetically heterogeneous clones can be derived from protoplast culture fusion, which display a high frequency of variation for several agronomical traits, and induction and control over the degree of specific chromosomes. Elimination in wide range of hybridization are possible. So that's it for this lecture. That was something to tell you about basically that how cybrids are produced. First of all, one protoplast is produced, then cybridization is done between two intergeneric species, or also species also, interspecies also, intergeneric also, intertribal also. So all these things were possible, and and uh, and you can do many things beyond that, like you can play around with the mitochondria, that you want mitochondria from one class and not from the other, you want nuclear from other one, not from other. You can play around it with anything that you like. So that's the best part of these hybrids thing. So, class, this is for today. I mean, I have one lecture also prepared for the hybrids, but this is all covered. I will send you these slides. They are the more easy version of it, but I have covered them already, everything. So like this diagram, it's not zooming. Just a second. I have to quit all to open a new. So this is another lecture that I have created, but most of the things are covered in this. Just want to show you this slide. That is, recipient cell line is there, there is a mitochondrial donor fibroblast, then you enucleated them, and then receptor cell lines are added with the mitochondrial DNA, then you have a cytoplasm fuse and select, then you have a hybrid. This is another way they have explained with the wild type mitochondrial DNA and mutant mitochondrial DNA. Just go through this part also. This is more easy version of it. I will also share this with you today, so I can share you now, I guess. 57 members. Okay, it is being shared. So any questions, class? So why protoplast method is applicable on the vacuolated cells? Yeah, because that is the only way we can transfer the generic terms, the genetic part to be fused together. Otherwise, with the uh, filled with the, yeah, otherwise it will be very difficult to go for. So any questions, students? Anything else? So it's not like that every day we have to go through 2 hours lectures, because I'm trying to finish this. I'm going through syllabus type. So we will be finished with the things on time. So don't worry that in one day, if I teach you everything, what will we be doing the last days? So don't worry, we will be doing, we are on time. We are doing good. So just go through the lectures before you sleep today. I think they will be sent to you today by soon. Most of them is all, yeah, they are sent. Just check them. I think I sent in the same group, and go through them each things. Yeah. Anything else, students? If not, then I would say good night. Take care. Then we see us tomorrow, same time, with more of next part of it. That is with the restriction enzymes and blotting technique. So we will be dealing that part tomorrow. That will be exciting. Maybe I will bring some videos to you for blotting technique for the Western blot, how you do it. So we will be dealing with that part tomorrow with southern blotting, northern blotting, western blotting, southern western blotting, northern western blotting. So we'll be dealing with all that part tomorrow then. Okay, on that note, I will say good night, take care, stay safe, and then, yeah, see you tomorrow. Have a blissful night then. Good night, bye.